Graham is head of translational science and experimental medicine operations for early respiratory and immunology at AstraZeneca. His role involves the development of the operating infrastructures to enable the precision medicine strategy across early respiratory and immunology projects. Graham is also the AZ lead for the 3TR IMI Consortium, a pan-European cross-disease research consortium aimed at improving personalised biological treatment of asthma, COPD, and leads the 3TR Breathomics Working Group. Thanks for joining us today, Graham, and we look forward to hearing from you.
Hello, my name is Graham Clarke. I'm the head of operations for translational science and experimental medicine for early respiratory and immunology at AstraZeneca, Gothenburg. And today it gives me great pleasure to introduce my presentation, which is addressing the utility of breathomics in drug discovery and development. Today I'll be walking you through how AstraZeneca is embedding precision medicine into its pipeline and what tools we are harnessing to identify the right treatment for the right patient. We will also be exploring how we address the changing target paradigm and how breathomics fits into that concept. And finally, we will look at the challenge ahead to breathomic methodology and new approaches which gives this technology promise for the future. Our bold ambition is to aim for disease modification and clinical remission in disease areas with great unmet need across respiratory and immunology. We are considered to be number one in respiratory with our innovative products and are widely recognized as having achieved scientific leadership. In column 1 on the left, you can see our core focus disease areas. Besides respiratory, we are now utilizing our expertise and knowledge to expand into the immunology disease area. And in our early R&I portfolio, we have emerging early stage assets aiming to treat immunological diseases with great unmet need. Moving to the middle panel, you can see how we're working to increase the probability of success for our molecules in a number of ways. Our ambition is to get away from me too targets in all comer populations by identifying differentiated and unbiased novel proprietary targets. And importantly, by utilizing our multimodality platform, we have the capability to address any target we uncover. No molecule is undruggable for us any longer. We also believe that in order to be successful, we need to treat the right patient with the right medicine. Therefore, we are applying a precision medicine approach from the start of drug development. In the third panel, you'll see how we are going to couple this with introducing transformative clinical endpoints, allowing us to capture disease modification in the clinic for characterization for our assets. This also includes analyzing clinical data in real time, using novel digital technology linked to the AZ digital platforms and assessing more patient-centric outcomes to add value for our patients. It really is precision medicine from the start. And when we talk about precision medicine, at AstraZeneca, we define it as an approach based on identifying the individual and measurable characteristics of patients in order to match treatments to patients most likely to benefit. And using diagnostics, biomarkers, and genomics, we can develop and deliver targeted therapies to patients. Why do we need precision medicine? Well, we need precision medicine because there remains significant unmet need in both asthma and COPD. Up to 20% of patients with asthma do not achieve an acceptable level of control, despite being prescribed high-dose anti-inflammatories and bronchodilators, and are referred to as having difficult to treat asthma or difficult asthma. In COPD, it's much the same. Even between exacerbations, when patients are considered relatively stable, symptoms are still prevalent and problematic. In the last 20 years, we have only discovered 3 novel mechanisms in asthma and 1 in COPD to effectively treat. We could be doing much better in this space. And precision medicine in AstraZeneca means targeting our innovative medicines to patients most likely to benefit. We have an opportunity to apply precision medicine approaches at every step in our portfolio, harnessing both data and samples from large patient cohorts in order to improve patient outcomes and broad access to new therapies. And when we talk about the changing the new target paradigm, what do we mean exactly? Well, we're deploying new unbiased novel target identification strategies, including the application of contemporary genomics to the identification of new drug targets, new patient groups, and new biomarkers for respiratory disease. Novel innovative approaches that will lead to new insights into disease biology and translate to greater success in the clinic for our respiratory medicines. High throughput technologies enable the incorporation of omics studies. Each omic layer allows a comprehensive survey of that particular type of disease association. We use multi-omic approaches to integrate data from different omic levels to understand their interrelation and combined influence on disease processes. Machine learning will identify novel patient subtypes from large, molecularly deep phenotyped cohorts. We now have both the data and the technical tools available. Generation of knowledge graphs to collect and connect all relevant data in one data schema allows for more unbiased exploration and assessment of current knowledge. Modern machine learning techniques, such as deep learning, can develop smart clinical phenotypes that can be used to find genomic drivers of respiratory disease. In fact, our success can be measured by 73% of AZ projects with target disease genetic links successful in phase 2 versus 43% without. The attraction, however, still lies with accessing the lung non-invasively. We can differentiate patient groups through the analysis, for example, of lung signaling cytokines like IL-6 in sputum. However, to deploy precision medicine globally at product launch, we will not be able to use lung tissue, bronchial biopsies, bronchoalveolar lavage fluid, sputum, for example. So we need to be able to access the lung without accessing the lung. And we're looking at different areas of research, notably around exosomes. So packages of lung-derived biomarkers found in blood, small RNAs, disease-specific new epitopes, so identifying and quantifying disease-specific protein fragments, nasal sampling, and wondering perhaps, does the nose reflect changes in the airway? Imaging, the application of new technologies to assess disease distribution in the airways, and of course, finally, breathomics, where we are seeing promising initial signatures which require the need for further validation. And so breathomics is important, both in terms of using this technology to target new biomarkers and discover new endpoints, but also for future diagnostic purposes as well. So target discovery or future diagnostic, where will breath analysis likely to have an impact? Well, exhaled breath contains thousands of volatile organic compounds, as we know, that reflect the metabolic processes occurring in the host, both locally in the airways and systemically. There is an unmet need for point-of-care respiratory-specific biomarkers that can aid in advancing precision medicine in both acute and stable respiratory diseases. Critically, they can be used as indicators of normal biological or pathological processes or pharmacological responses to therapeutic intervention. But also early, rapid detection and treatment of lung diseases can improve patient care and adopt a more personalized therapeutic approach. It can also be used in longitudinal monitoring. So can we use this methodology to detect exacerbations or treatment response? And perhaps lastly on this slide, a consideration of how and where we deploy it. Where could and should breath analysis be used? Moving it from the hospital to the clinic, to the GP, to the home. So the potential for using home care testing is very attractive for point of care tests. And breathomics has an even bigger opportunity to explore different disease phenotypes. So can breathomics be used to address the unexplained heterogeneity of disease in asthma and in COPD, or indeed other disease areas as well? The number of breath biomarker discovery trials has increased in recent years, particularly in asthma, where there's been 3 published systemic reviews by Rufo, Peel, and Asim. And it confirms the discovery of potential breath volatile biomarkers across multiple studies. But none of the findings to date have been replicated in independent prospective cohorts. But the adoption of breath into large-scale and multi-platform studies such as Biopsy, shown here on the right, MRC, EMBER, and most recently, the broad European initiative IMI 3TR, confirms there is interest in breathomics. And as a potential point of care tool, VOCs have been linked to urinary traces of salbutamol and oral corticosteroids, and suggest that breathomics may qualify for development into a point of care tool for monitoring asthma drug level changes, as noted here in the European Respiratory Journal last year. So what are we measuring? So this figure looks a bit complicated, but it really highlights the complex kinetic of gaseous exchange. We have endogenous VOCs, which originate from the lungs or distant organs via systemic circulation. Exogenous VOCs are continuously introduced into the respiratory system, which can result in the production of volatile downstream products by various interactions with airway microbiota and mucosal lining. Breath samples containing endogenous and exogenous VOCs are analyzed to generate clinically meaningful data. So in asthma, it has been shown that a broad spectrum analytes can be measured, including carbonyl containing groups, i.e. aldehydes, esters, and ketones, and a broad selection of hydrocarbons, i.e. alkanes, alkenes, and monoaromatics. However, there are challenges. The removal of exogenous VOCs may simplify analysis, but loses potentially useful signals. And two, the analysis of environmental exposomes needed to give certainty of VOC origin. In this slide, it highlights a paper that addresses the longitudinal changes between clinically stable episodes and loss of asthma control, and with it, a number of potential biomarker candidate VOCs, synergistic with clinical endpoints. Here I've highlighted 3 VOCs and their composite PC1, which forms the possible correlated outcome analysis in this paper. In this paper, 23 patients with partly controlled mild to moderate persistent asthma using ICS were included in this prospective steroid withdrawal study. Exhaled metabolites were measured at baseline during loss of control and after recovery, and standardized sampling of exhaled air was performed, after which samples were analyzed by GCMS and ENOS. A composite of VOCs were identified, that's PC1, and their expression measured against blood EOs and neutrophils, sputum neutrophils and eosinophils, FEV1, and exhaled nitric oxide. There were other parameters as well which were measured, but I'm just going to focus on these 4 points in the top right of this slide. But unfortunately, there was no correlation in blood, but there was positive correlation of these VOCs with sputum, eosinophils, and FEV1. And one component of the PC1 composite was bicyclo-2, and that was correlating rather well with exhaled nitric oxide. On the bottom right hand of this graph, it shows that the associations between GCMS PC1, that composite of these 3 VOCs, and log transformed sputum eosinophil percentages by ANCOVA analysis. And there was a strong association, and that was significant in 16 out of the 22 subjects, based on 3 successful sputum inductions at baseline, loss of control, and recovery. So this paper creates hope, perhaps, that breathomics is somewhat synergistic with clinically stable episodes and loss of asthma control. And in this slide, we see some insights into COPD and smoking. In this paper, we see a summary and identified here 7 out of 10 novel breath VOCs that this paper identified related to COPD, using a different approach in breath sampling compared to others, and by using a limited number of target ion identified VOCs. This group in Germany confirmed the profound effect of smoking in their study and show that so far unexplained site-specific differences do exist that need to be taken into account when interpreting results from multi-center studies. But if we assess the importance of VOCs in smoking and COPD, these VOCs show a difference between controls and COPD patients in all subjects. In subjects of each site, and there are the open circles from Hanover and the closed circles from Marlborough, and in non and ex-smokers, which is on the left, and smokers, which is on the right. In this next slide, we identified, but there are inconsistencies. Whilst there are several studies identifying VOCs correlating with established pro-inflammatory indices, there are others which highlight no relationship at all. Results from the published Alliance cohort highlighted a group of patients prospectively recruited. These were 133 adult patients with diagnosis of asthma across different severity grades and inflammatory phenotypes, as identified by exhaled nitric oxide, blood and sputum differential cell counts, and breath VOCs were compared between different asthma phenotypes as well. But unfortunately, there was no correlation across the parameters with any VOC, with blood eos and neutrophils, sputum percentage eosinophils, pheno, and FEV1. And it asks us the question, are we doing this right? How close are we getting to finding a clinically valuable breath VOC biomarker that addresses the disease heterogeneity which exists in asthma? So we have a challenge ahead. In this slide, we look at the challenge ahead. This is a nice review from Brinkman in 2019. It was titled Breathomics and Treatable Traits for Chronic Airway Diseases, and it summarized three main categories of causes that hamper the valorization of breathomics in clinical tests, including recommendations and potential workarounds. And they are heterogeneity of study populations, diversity of hardware, and differing models and reporting strategies. And under the heterogeneity of study populations, we need to look at lifestyle, age, comorbidities, microbial processes, and medication use. Under diversity of hardware, we need to look at mass spec versus sensory-driven techniques, real-time versus offline analysis, direct sampling versus sample preparation. There are differential detection limits which might be applied using different technologies, and of course, predefined versus untargeted analysis. And then finally, in the differing modeling and reporting strategies, we need to be far more consistent in how we deliver this message. Is it data-driven or hypothesis-driven analysis? There are differences in pre-processing steps between the different technologies which we need to standardize. And we need to make a decision on what statistical approaches we need to take using univariate or multivariate outcomes analysis. We need to look at inclusion, exclusion, external or internal validation steps. And are there sufficient guidelines available now to determine the use and deployment of these technologies to measure breath VOCs? And it's complicated. So there's a bench-to-bedside patient stratification approach that could be based on molecular disease processes that we could use. So transcriptomics is perhaps our example. So where do we go from here? How do we begin to find our target? Well, if we're able to use transcriptomics as our example, it tries to address a bench-to-bedside patient stratification based on key molecular disease processes. With a cell-based model, we can move to relevant disease-related stresses to addressing a response and biomarker gene signature. We can observe these changes in disease and define a patient subset by disease mechanism. In the example used here, of course, we have determined local lung epithelial IL-6 transsignaling activation in the absence of type 2 airway inflammation, defining a novel subset of asthma patients which might drive airway inflammation and epithelial dysfunction in these patients. For breathomics, what might our first step look like? The hypothesis, well, the lung epithelium is a major source of VOCs in exhaled breath, and VOCs in headspace air collect from lung epithelial cells in vitro may provide insight into the in vivo production of VOCs and thus the exhaled breath composition. The suggested approach might be that we need to establish VOC patterns in headspace air, collect from lung epithelial air liquid interface cell cultures treated with different disease-relevant stimuli, and then identify stimulus-specific VOCs patterns in exhaled breath from patients and explore utility for patient segmentation based on biological drivers of disease. But it's not easy. So connecting the VOC biomarker to the underlying biology has its challenges. Connecting the VOC biomarker to the underlying biology is difficult. The opportunities are manifold. Cellular and tissue profiling is an enormous task and difficult, but perhaps through the use of layered omics, we can walk in the right direction. But the inescapable challenge would appear to be the understanding of where the VOCs come from. How do we understand VOC origin, both endogenously and from the environment? Firstly, the technology is improving. to remove the pollution of exogenous VOC from the environment using Casper filters provided by Alstone. Secondly, where does VOC come from endogenously? Well, Alstone are developing an EVOC probe method as a reliable and highly sensitive alternative to untargeted biomarker discovery. And this approach offers the advantage that compounds not normally found at significant levels in breath can be introduced into the body individually or as a cocktail to explore how they are absorbed, metabolized, or excreted. The concept involves exogenous VOC probes, which are safe compounds that, when administered, undergo a metabolism in the body and are excreted by breath, offering a readout of metabolism by enzymes and organs. This approach could permit high levels of compounds to be administered, substantially improving signal-to-noise ratios compared to signals from endogenous sources, and thus greatly improving reliability of detection. And we see other groups addressing this challenge. The rate-limiting step in the translation of breath volatiles to clinical diagnostics is the identification of disease-specific breath biomarkers. So in this paper from Chan et al., entitled Engineering Synthetic Breath Biomarkers for Respiratory Disease, they deploy a schematic to highlight their approach. So in A, on the left-hand side of the schematic, a VOC-modified peptide substrates are formulated into a volatile releasing activity-based nanosensors known as VABNs by conjugation onto an 8-arm PEG nanocarrier and delivered into the lungs via intratracheal instillation. In B, when attached to the VABN, VOC reporters are in an undetectable non-volatile state. That's the gray cloud around that middle section. Extracellular proteases produced during the respiratory diseases cleave surface conjugated peptide substrates, thereby releasing VOC reporters. Upon release of VABNs, reporters recover their characteristic mass and volatility, and that's the orange squiggly lines, and are then exhaled. In C, towards the right-hand side, breath is collected as perhaps in other techniques in a receptacle, like an evacuated glass vial, and VOC reporter concentration is quantified using mass spectrometry. In short, it's a bottom-up approach to addressing where we confidently are able to source these VOCs in the airway and gives us some confidence in addressing the underlying biology. So I'm coming towards the end of my talk, and we want to address what the immediate focus of summary should be. Where do we go from here? But the immediate focus for where we take breathomics is important to ensure its future sustainability in addressing point-of-care diagnostics, at the very least. Can breathomics be deployed to address the unexplained heterogeneity of disease in asthma and COPD? That remains to be seen. There are encouraging signs in the data on certain VOCs being identified and correlating well with clinical endpoints. But there is a need to focus on sample preparation, experimental analysis and quality control, metabolite identification and data processing applied across all breathomic techniques in order for us to diagnose a patient's condition and match that diagnosis with medicine or therapy. There's an awful lot to do in terms of proper application of pre-processing steps, the optimal choice of analysis algorithms, and rigorous validation of machine learning. Precision medicine offers the promise of quickly and unambiguously diagnosing a patient's condition and matching the diagnosis with a medicine or therapy determined in part by the genotype or phenotype. Breathomics offers an opportunity to meet these requirements. There is an opportunity to harness the power of multi-layered omics to determine meaningful targets and patient phenotypes, and how breathomics synergizes with these approaches remains to be seen. There are encouraging developments in substantially improving signal-to-noise ratios compared to signals from endogenous sources, and thus greatly improving reliability of detection. But what is critical is the sharing of data and the transparency of method development across both laboratories and clinical sites. we're comparing different exercises and approaches with a wider consortia of individuals. I represent IMI 3TR. This is a consortium which looks at, broadly speaking, the use of biologics in patients who don't respond necessarily to conventional therapies. And I'm heading up the Breathomics Working Group. And this interaction between industry, academia, and the breathomic industry is critical to driving better standards and insights in the deployment of these new tools and techniques. So I'll leave it there with you today. Thank you very much for listening, and I'll be happy to take any questions. Thank you.
So first question I had related to the relative likelihood of success of endogenous VOCs versus some of the voc probe candidates. So we have Melody from MIT speaking at the conference tomorrow about some of the protease work that they had previously published. I know it's pretty early days, but do you have a bit of a sense of where success is most likely to come, endogenous voc probe, or is it going to be a mix of both, in your opinion?
Probably a mixture of both, Billy. I think it's an exciting point for this technology. One of the biggest headaches, and I think some of your questions in the chat might reflect that, is understanding how we tease out which VOC is important for which therapeutic area, which disease process. The underlying biology is critical for us in certainly in AstraZeneca in defining certain targets. So these probes might give us some confidence in establishing which target tissues we should be focusing on, and indeed how we discriminate between one cluster of VOC biomarkers versus the next.
Fantastic. I know there's focus primarily on VOCs. Obviously, breath has exhale breath aerosol as well, so you can think about some of the non-volatile components there. I wonder if you have any thoughts on the potential utility of exhale breath aerosol and some of the compounds or the biomarkers that may be found there for sampling deep airways without having to do things like BIO.
Yeah. So there's great interest in this area. The aspirates or localized to remove more local sampling, and often these things, Billy, it may well be a composite of different techniques which are able to bring together a map of the airway. I think the challenges we have, and imaging helps us to some extent of where we can look at the geography of certain disease process. But we're hoping perhaps there's certain flow rates or the aspirates we sample give us some indication. I'm hopeful. I don't have much information in that area myself, but I'm very hopeful that the technologies required to assess where these aspirates, what's in these aspirates and where they're from, gives us some potential insights in how we might use this technology in the context of other assessments.
The next question from Scott. Do you feel the current disease classification by symptomology hampers research in breathomics? I think you kind of touched on that a little bit, the difficulty of getting to ground truth in terms of the label.
Yeah, I think what we've know certainly with asthma and COPD, and I mentioned it in the presentation, is how do we look at unexplained heterogeneity, and that's thinking beyond the symptomology as well. Symptoms are important in how we monitor treatment and resolution, but it doesn't necessarily help us in differentiating one phenotype versus the next, particularly. And I think we have some way to go, but breathomics might have that opportunity.
Fantastic. There's a question from Hendrik. You said earlier that breathomics can also be brought to the point of care at the customer's home. What are the current approaches? What are the biggest hurdles from your point of view?
Yes, so I mean, it's nice to have that vision, isn't it? I think certainly the last couple of years has really focused the industry on how we monitor patient care and how we make it easy. And and what it is that we might lose. I think for these technologies to evolve, they have to almost be bulletproof in its deployment and utility in the hands of the untrained. And I think it has enormous potential in how we perhaps might standardize certain sampling techniques. But we don't know yet. I'm sure, Billy, you're working on the technologies to standardize the environmental components of sampling at home. But I think the hurdles really is that standardization piece, the reproducibility piece, and the ease which these things can be used by the untrained.
Well, one follow-up question I had that related to that, be interested in your perspective of how important it is from the point of view of sample collection in the home versus the turnaround time on the result. It feels like in some instances you get a big advantage if you can collect the sample in the home-based setting, but lab-based analysis may be acceptable. But in other applications, actually it's the turnaround time. They get the longitudinal data that becomes very powerful. So it'd be interesting in perspective on those subtleties and differences within respiratory disease.
I agree. Yeah, absolutely. I think, and then I think, Billy, it may well be that certain clusters or combinations of volatiles or other markers might be less sensitive to the sampling path, I guess, or transition than others, and that's something we're very keen to understand. The storage of these samples and what happens to these samples in preparation over time.
Yeah, and I think that's interesting as well. Like with the vocal probe strategies, one of the big advantages you get is you can pick what your reporter is, make it unique, make sure that the storage is better compared to some of the other compounds as well. So I think that's another important advantage. Question here from Andrea. Thanks so much for the nice presentation. What are your thoughts about the biomarkers that are common between various respiratory diseases?
Yeah. So this comes back to origins, and I may not be able to answer this question directly because I think on its own it's quite challenging, but it's all within the context of the patient. And one of the things we're very keen to understand in the breath analysis is how well do you slice that cake between what is asthma and what is something else? And I think that's the biggest question we have in the selection of biomarkers, which may be specific to an individual or an individual phenotype or the disease. And that's the exciting prospect of where we look at the analysis is where we slice. And how meaningful some of the data is sensitive to either treatment or longitudinal understanding of their disease process. So I think in my mind, the jury's still a bit out on that one.
Great. I think we have time for one last one. So you touched on it a little bit in the presentation, but I just wonder if you could elaborate a little bit further into to what extent in vitro studies can help inform in vivo breathomics studies. What's your kind of view on that?
Yeah. So I was trying to use the transcriptome as a look-see at how we might establish that. I think, and I mentioned, of course, that cell and tissue profiling is actually quite a big task, and I think we need to be far more discriminating and maybe take a more high-risk approach with some of these methods. I think we have to be very clear that we still have the line of sight to the clinical endpoint, and that's where I think we're trying to build that gap. So what we're measuring in the breath and how meaningful that is to the clinical phenotype, and somehow bring it to the biology. But we have to, I think, in order for us to validate the target, we have to do some of these in vitro studies, understanding more about the metabolome in particular, and how all of these layered omics, where does breathomics sit in the plethora of omic approaches, going from the genome, which we've mentioned before, all the way to the breath, I think for us, every individual has the potential to have this layered makeup map of their own personalized disease profile. That maybe breath on its own may not be enough, but it certainly is a strong component of how we address it.
Yeah, absolutely. Make those integrated studies all the more important at the outset. Well, Graham, thank you so much for the time today. That was a really fascinating talk, and really appreciate you joining us. Have a great rest of your day and enjoy the rest of the conference.
Thank you, Billy. Thank you.